Hello, everybody. Mr. Andreyko here from my basement. I've been projecting lots of lectures of biology from the basement, but not so much for forensic science from the basement. Unfortunately, forensic science is very much a lab and textbook course, and since we didn't have enough textbooks and we don't really get to do a lot of labs, this course is kind of limited in its online form. So we'll move through the material. I'm going to set you guys up with a project this week so that you guys can, work on a little bit, learn a little bit, not be overwhelmed with your other classes, this being an elective. So I'll be taking it easy on you this week, but you will have an assignment from me coming up in the next day or so. So be on the lookout. It is related to genetics, DNA, and how it is related to forensic science. The purpose of this video is to walk you through genetics and DNA, a quick run through what you learned hopefully in biology, either by me or your biology teacher, to give you the basis of how that applies to forensic science and how it's used in a court case, obviously. So since DNA is the most important, we've kind of saved it till last. And so let's get to it, right? Genetics is the study of your genes, your inherited traits, and it is based on your DNA. DNA is found in every cell of your body. It is the code that makes the instructions for you, aka the blueprints for life. DNA is made of just 4 different base pairs, adenine, guanine, cytosine, and thymine. And they pair up A with T, C with G, right? Hopefully you remember this stuff. Maybe the term nucleotide rings a bell. Anyways, this is the basics of genetics and the structure of DNA of what you should have kind of taken away, right? This base pairing. And these letters, right, they code to make proteins, and those proteins are used for muscles and bones and cell parts, but as well they're for enzymes and antibodies and other proteins that do stuff, right? So they give you the form and the function, and all of that comes from the boss, which is DNA, this blueprint. So we've all got to have plans of how we were built. Now, most of DNA is similar in all living things. We share lots of DNA even with bacteria, corn, pigs, one of the closest species right now that we're actually using to help us with the coronavirus testing, specifically on the vaccine side, is the rhesus monkey. And so we are 99% similar, well, depending on, how you blow up that data, but we are very similar genetically to those things. So you can imagine 2 humans are even closer genetically, right? We got the same number of chromosomes, right? Each got 46 chromosomes, and through meiosis, we produce 23, and they match up, right? That's how 2 species are identified. And so our DNA is very, very similar. So how is it that we can actually identify one human from another when most of your DNA is the same? Well, luckily, DNA is written kind of like a book, and so it has spacers in it. I try to use the examples of, like, when you finish, like one section, say, of some questions, right? I'm done with the questions, and I'm ready to move on to the review in a book, right? And so you just leave the page blank, and then you go on so that the next thing is on this page, right? And so your DNA does the same thing. So instead of a blank page, it just has junk DNA. And so that junk DNA is what is unique to you, right? So These are the 13 loci. Loci is a plural word, right, for locust, not like locust, like the bugs that fly around in the desert, but locus is a specific location on a gene or on a chromosome. And so some of these code for your eye color, how big your ears are going to be, what your hair color going to be, are you going to, end up bald like Ann Draco, right? All of that is written in here. But sometimes there are spacers, like I showed you. And so we've identified those 13 spacers as junk DNA. And so they're any letters. And so those are the letters that are unique to you. And so we figured out how to isolate those 13 spots and look at them to identify who's different from who. How do we do this, right? Well, first thing we got to do is we've got to get the DNA out of the cell, right? You've seen in all the TV shows, most of the time they do what's called a buccal swab, B-U-C-C-A-L, where they go in your buccal cavity and they scrape the inside of your cheek, right? It's easy to get cheek cells off of your body, but whatever's found at the scene, blood, hair that has been forcibly removed, semen, things like this, all of this stuff can be used as a source of DNA. We spin it in the centrifuge because that DNA is the most dense part of a cell, and so it is pushed to the bottom through that centrifugal force, and then we just extract just the DNA. So now we've got concentrated DNA, and we want to find those 13 loci. And of course, good old enzymes are there to save the day, specifically called restriction enzymes. Restriction enzymes go in and they find those 13 spots and they cut those spots out and they allow them to be analyzed. Those fragments of DNA that we cut, they travel through the gel, right? And this is where the electrophoresis part comes in, is where the block is not a solid block. It's more like, say, Swiss cheese or a sponge where it has a bunch of holes in it. And the DNA goes through it like a filter. And so the electricity, or electrophoresis in this case, is what is running the current that is pulling the molecules through the gel. And so the big pieces get stuck on one end, the little pieces make it to the other end, and then we can analyze. And so as you can see, right, once we mix the enzymes in, we put them in, we turn on the current, we use the gel itself as a filter, and then we can look at it through UV lighting, and we can start to match. This is what is called its banding pattern. And so we match its banding pattern, and then we can see if things are related, if they're not, potential suspects. Of course, one of the projects you guys are going to work on will be people who have been exonerated due to DNA evidence. We'll look at the happier court cases. And so DNA has exonerated many wrongfully accused, so we're happy for that. Sometimes if bodies are unrecognizable due to catastrophes, car accidents, you name it, we can identify bodies that way. Of course, everybody has seen clips of Maury Povich, you are not the father, right? And so we can use gel electrophoresis to identify paternity or other relationships that people may know. There was a president I was just reading about who a woman claimed to have given birth to one of the president's bastard children, and they wrote her off as a whore and a liar, but later they did DNA testing to prove that she was in fact telling the truth. And so that happens as well. We can look at identify endangered and protected species, people who may have, parts or, claws or fangs or whatever of endangered species. This isn't as big a deal around where we live, but other parts of the world and maybe even other parts of the country are more interested in having endangered species. And so to identify them, we use gel electrophoresis. A big one is nowadays we have a lot of problems with contaminated food, uh, water, uh, things like that with bacteria, right? And bacteria is a living organism. Same thing with like Giardia, other protists, right? That can cause digestive problems or other health problems. And knowing what species of bacteria it is, we can figure out, did it come from the soil? Did it come from the dirty animals? Did it come from, right, the air, the water? And so identifying bacteria can be very helpful in trying to keep our air, our food especially, much cleaner. We can use it obviously to match donors. Um, Pedigrees for livestock. This is actually not to figure out if they're related, it's to find out who is the least related, right? Because as we've learned, inbreeding bad, right? Hybridization good. And so we like to try to determine pedigrees to make sure that we're not breeding anything too closely related. Even with wine and other consumables, they like to do this. That's kind of a very rare one. Um, but of course, your biological tie in, we can learn more about evolutionary descent as we find fossils and other types of evidence. We can use that kind of information, if there is still DNA that is viable, to help us with learning more about our evolutionary descent. So, that's it for your lecture on DNA in forensics and how it applies in court cases. The rest of this unit is you guys actually looking at court cases that involve DNA, because this is the bulk of what most court cases come to, that end up with some kind of resolution, because DNA is most of the time the definitive piece of evidence that we're looking for. The rest of the stuff that we've been looking at has been corroborating evidence, just helping to create or at least look at the recreation of the crime scene. This is where we can actually go from class to individual. So, so that's it, and keep an eye out for other stuff. Seniors, real sorry. That's all I can say. I've been thinking about putting out a separate video for you seniors. I'm sorry you don't get a normal senior year. I hope that you end up having a good summer, or college treats you well, or you get a good job, or you take a nice long vacation or something, and you enjoy it, because I'm sorry, as word has just come out, that the rest of the school year is canceled, and I feel for you. So that's all I got to say. Andre, go out. You'll hear from me soon.